Now we'll discuss about protoplasm. Parts of protoplasm, nucleus and cytoplasm. Nucleus, it is the control center of the cell. Every matter that happens within the cell are controlled by nucleus. Nucleus consists of nuclear membrane, nucleosome and nucleoplasm. Nuclear membrane are double layered, outer layer and inner layer. Outer layer is rough and it is attached with ribosome, and outer layer is continuous with rough endoplasmic reticulum. Inner layer is smooth. There are gaps between outer layer and inner layer, and this is called nuclear pore. Substance are exchanged between nucleus and cytoplasm through this nuclear pore. Nucleosome consists of DNA, chromatin, nucleolus and chromosome. DNA, deoxyribonucleic acid. DNA consists of sugar, nitrogenous base and phosphate. Sugar. DNA contains deoxyribose sugar. Deoxyribose sugar C5H10O4. This is the structure of deoxyribose sugar. This deoxyribose sugar comes from glucose. How it comes now we'll discuss. Glucose C6H12O6. Oxidation in glycolysis, it converted to pyruvate plus ATP. Glucose oxidation in HMP shunt, hexose monophosphate shunt or pentose phosphate pathway. In this pathway, glucose is converted to ribose plus NADPH. This is ribose sugar. This is deoxyribose sugar. The both structure are nearly same. Ribose sugar in 2' position, here is hydroxyl group, but in deoxyribose sugar in 2' position there is hydrogen. It means ribonucleotide reductase enzyme, when replace hydroxyl group of ribose sugar with hydrogen, then ribose sugar converted to deoxyribose sugar. Then what we understand? Glucose by HMP shunt converted to ribose, and ribose converted to deoxyribose sugar. DNA also contain nitrogenous base, two types, purine and pyrimidine. Purine contain adenine, guanine. Both are double ring structure. Pyrimidine contain cytosine and thymine. Both are single ring structure. When one sugar, either ribose or deoxyribose sugar, bind with a nitrogenous base, either adenine, guanine, cytosine or thymine, it produces nucleoside. Sugar plus nitrogenous base form nucleoside. This is sugar, this is nitrogenous base. There are so much position, there are also so much position where they will bind one another. To know this, numbering of the ring is important. In sugar, only carbon atoms are numbered with prime number 1 prime, 2 prime, 3 prime, 4 prime and 5 prime. In nitrogenous base, both carbon and nitrogen atoms are numbered. In purine, there are two rings. In first rings, it is numbered anti-clockwise. 1, 2, 3, 4, 5 and 6. In second ring, in purine, it is numbered in clockwise. 7, 8, 9. 1 prime carbon of sugar always bind with 9 position nitrogen of purine and form either adenosine or guanosine. Pyrimidine, it is a single ring structure numbered in clockwise direction. This is 1, 2, 3, 4, 5 and 6. A pyrimidine, nitrogen of 1 position, always bind with carbon of 1 prime position of sugar and form either thymidine or cytidine. Sugar plus adenine form adenosine. This is a nucleoside. Sugar plus guanine, guanosine. This is a nucleoside. Cytosine plus sugar. This from cytidine. This is a nucleoside. Thymine plus sugar. This from thymidine. This is a nucleoside. DNA also contain phosphate. Phosphate bind with sugar at 5' position and form nucleotide. It means phosphate plus nucleoside is equal nucleotide. Adenosine plus phosphate, adenosine monophosphate, it is a nucleotide. Guanosine monophosphate, it also a nucleotide. It also a nucleotide, it also a nucleotide. All are nucleotide monomer. One nucleotide monomer can bind with next nucleotide monomer by 3,5 phosphodiester bond and form polymer of nucleotide, and this is called DNA. This is single stranded DNA, and this type of DNA is found in single stranded DNA virus.